Okay, hello, welcome. Please come in and take a seat wherever you can. So welcome to Learning and Memory, How Experience Changes the Brain. I'm Karen Campbell. I'm from Brock University, and we've got Dr. Rosanna Olson from the Rotman Research Institute in Toronto. And we're happy to have you all here today. So our first speaker is Leah Tolazzi from the University of Bordeaux. So please come on up.
Hi. Yes, I'm Leah Tolazzi. I'm here to work with you my current work in a latent disconnectome prediction of long cognitive symptoms in stroke. A stroke occurs where we have a cerebral vascular injuries in the brain. As a macro categorization, it can happen in the posterior, middle, or anterior cerebral arteries. Thanks to magnetic resonance images, it's possible to delineate in vivo the lesions with a quite good accuracy. Here you have an example of two different waves that we can have in MRI, and with the red arrows you have pointed the lesion location. The goal of our project was to associate the stroke lesions, and in particular the white matter distractions, with clinical symptoms. For doing so, we delineated the stroke lesion in each patient. Then we used the normative data set from the Human Connectome Project, acquired 7 Tesla, where we reconstructed a whole brain tractography that served for filtering, thanks to the stroke lesion, and obtained the stroke disconnectome, so the amount of white matter distraction that we have in each patient. Then, to deal with the variability of the disconnectome patterns, we used dimensionality embedding techniques. Then we wanted to correlate the neuropsychological scores with this location in the embedding space. Then we bring it back, this information, in the white matter, and we obtain an atlas of neuropsychological symptoms. And then we also released a web application for running online single patient predictions. Here, the codes that we used. The first was acquired at UCL London, a big data set with more than 1,000 patients. The second and the third one was acquired at Washington St. Louis. So all the MRI was acquired within 1-2 weeks from the stroke onset. And for data set 2 and 3, we also have 86 neuropsychological scores. Keep in mind that the neuropsychological scores were acquired at 1 year from the stroke onset. So we used MRI scans acquired between 1-2 weeks to predict long-term cognitive symptoms at 1 year. Then we use the Uniform Manifold Approximation and Projection method, UMAP, for running the low dimensionality embedding, and so to map the variability of the disconnection that may occur for all these patients' population. We run the UMAP in the first dataset 1, the biggest, and we defined what we call the disconnectome morphospace. So here, 3 patients with different disconnectome patterns, and you can see the projection in a 2-dimensionality space, and so the coordinates associated to these 3 patients. These serve as a reference framework to import new patients. Here you have an example of dataset 2 and 3 imported in the same dataset 1 framework. And you can see how patients with similar disconnectome patterns are projected close by in the disconnectome morphospace. The neuropsychological scores acquired at 1 year were 86. Here, a subdivision with the functional domains, so for motor function, language, visuospatial ability, visuospatial memory, verbal memory, sickness, and pain. We imported these scores, we associated them with the coordinates in the morphospace, creating a composite morphospace where we have also the information of the different neuropsychological scores. Here an example with the left hand grasping, and you can see how the lowest score, so when we have an impairment in these particular scores, are well segregated in a specific risk territories in the embedding space. We run correlations, peak pixel-wise in the morphospace. And here I reported the correlation with R in module higher than 0.2. And we used these risk territories to run a principal component analysis and predict the long-term neuropsychological scores. Then we wanted to bring it back, this information, in the neuroimaging space. And so we ran correlations with the different disconnectome patterns in the neuroimaging space, and we obtained effect size maps. Here is a summary. You have the different acronyms associated to the 86 neuropsychological scores, colored by the different domains, functional domains. In panel B, you have the highest effect size achieved. As you can see, the highest effect size was in the left frontal lobe white matter projections. And then the versatility map, where we can appreciate the overlap of the different neuropsychological scores, and we had a higher overlap in these 89 neuropsychological scores in the right hemisphere. Then we use dataset 3 as an out-of-sample validation. Indeed, the regression formula was trained in a dataset 2. Here I reported 2 patients, the first with an impairment in the right motor functions, and the second in the language domain. We run the prediction formula for each patient, and on average, we obtain a maximum prediction mean absolute error of 16.1 on average. Then, as anticipated, we developed a web application. Here, I leave you the QR code where it's possible to run the online predictions. Here, you have a summary of how the model works, so the different neuropsychological scores, the acronyms that I showed you before, and their explanation. And going to calculate, you can then upload a single patient disconnectome file. You can also download an example in the web application. It will be registered in the MNI space 2 millimeters. It takes less than a minute, and you will see the UMAP coordinates here, and then the predictions scores here for the motor functions in the left and right abilities. And then you can also download the file as a CSV file and have it locally. So in conclusion, we use dimensionality embedding techniques to map the variability of the disconnectome patterns in our disconnectome morphospace. That serves as a framework to predict long-term cognitive symptoms in the stroke patient at 1 year. Then we create a white matter atlas where we correlated the findings in the embedding space with the neuroimaging space, in particular in the white matter. And then we released a web application that serves as a broader validation of our model and to really get tested this model. So you can go through the QR code or is in the www.disconnectomestudio.bcblab.com. I would like to thank you for the attention and all the collaborators of the project, Michelle, Stephanie, Valentina, Victor, Parashev, and Maurizio. Thank you, and I'm happy to take your questions.
Okay, so we've got some questions coming in on the app, but if you prefer to ask your question in the room, you can just put your hand up and we'll bring the microphone to you, okay? So the first question is, could you tell us a bit more on how the 2D UMAP is derived? Thank you.
Okay, so the UMAP is a non-linear embedding method. So, for example, as a contrast with the principal component analysis, that is a linear method, in the non-linear method, you really look to find variations. So you use mathematical tools as the Laplacian model to really look to the variation. And these give you a map, so a Cartesian space, where you can plot the disconnectome variability. So this serves as a framework to do a low dimensionality reduction. So you pass from the imaging space that are a lot of voxels in 3D to a 2D. So it's really a big reduction. Mm-hmm.
The next question is, could you tell us a bit, oh, sorry, that's the same one. Besides the original lesion, what other variables were accounted for in the production of this model?
Sorry, how the lesions are derived?
What other variables did you account for in the production of the model, apart from the original lesions?
No, we just used the lesions for the patients, and as I show you, we used the Human Connectome project, DTI data acquired in 7 Tesla. So the cartography was derived from the Human Connectome project, and then it was filtered with the stroke lesion. That is really useful because then you don't need an advanced acquisition, MRI acquisition for the patients, but just an T1 and T2 MRI scan, and then using our software and the VCB Lab toolkit to derive the disconnectome, you can run these sophisticated models starting from just the lesion segmentation.
So another question is, how is it possible to project the neuropsychological data into the same UMAP space?
So as for each patient, you have 2 coordinates, and the same patients have associated the neuropsychological scores. So this is basically the association, so the single patient. And then as an independent variable of the regression formula, you have the neuropsychological scores. And as a dependent variable, so the input of your model will be the composite morphospace PCA components.
I think we have, no, I think that's it for
okay,
time.
the time.
Yeah, thanks.
Okay, thank you.
Okay, we'll welcome our next speaker to the podium. Our next speaker is Joana Carvalho. She's from the Champlamode Foundation in Lisboa, Portugal.
Good morning, everyone. I'm very happy to present to you how visual experience can actually shift the stability-plasticity balance in visually deprived rats. I'm well aware that we are at the Human Conference Mapping, but I hope throughout the course of this presentation I can show you how can we link preclinical data to human, in both in terms of the techniques that I applied and how can we validate them, as well in terms of the generalization of the findings. So in human and non-human primates, fMRI experiments normally harness visual stimuli to actually map the retinotopic organization of the cortex, both in terms of position and also in terms of the size of the population receptive field. However, such mappings are not possible in rodents, where we can have in-depth biological information due to manipulations of vision vis-a-vis plasticity, and also due to orthogonal validations using histology. So for now, in rodents, the mapping of the receptive fields and the retinotopic structure is limited to electrophysiology and calcium imaging. And because our main goal is to actually know if we can map the large-scale topographic remapping in the visual pathway, and to know if it can take place in visually deprived animals and then be shaped by visual experience, first we had 2 main goals. The first was to actually be able to characterize the structure and function of rodents using fMRI, so in a non-invasive way, and to use this detailed characterization then to understand this dynamic time course of the stability-plasticity balance and how it can be shaped by visual experience. So our first challenge to actually be able to perform this is how can we actually display an image inside preclinical scanners which have a very, very small bore. To do that, we built a customized setup, and together this setup with a specially designed stimuli to perform spatial frequency turning curves. So I presented multiple spatial frequency gratings and retinotopic mapping. Basically, we have a visual stimuli that swipes the entire visual field. We wanted to study how the brain function and structure is altered between normal animals or that grew up in normal conditions and visually deprived ones. And when I talk about visually deprived animals, I'm talking about animals that were born in the dark and never saw light until the first time they were scanned, which was at time point 0, and they were already considered adults. Then we scanned then after the first time that they were scanned, which was also the first moment of light exposure, the animals were placed in the normal environment of 12 hours day cycle and 12 hours night cycle, and they were scanned 1 week after, and at 17 days after light exposure and 27 days after light exposure, and we compared their topographic mapping with the one of healthy controls. So we scanned 10 dark-reared and 10 healthy controls. The animals were scanned at a 9.4 Tesla, and we scanned at a very nice spatial resolution. So just to tell you, using this setup, we could actually map the receptive fields at 125 by 125 microns throughout the entire visual pathway of rodents. And just to make you a little bit of introduction, a receptive field corresponds to the position in the visual field that the neuron responds to, and the rat visual pathway is basically is composed by 3 main components. So the information comes from the visual field, crosses at the optic chiasm, and is then processed at the LGN, superior colliculus, and then at the visual cortex. And these are the 3 main areas that we are showing here with a very nice retinotopical organization following the visual field map. Then we looked at the PRF size, or the population receptive field size, and how it varies across cortical depth. We can see that the receptive fields are the largest at the most superficial and deepest layers, and the smallest at layer 4. And actually, this is very well represented when we actually can follow the voxels across one cortical column, and we can compare these with the electrophysiology, and we see actually this gradient from larger receptive fields to very well-defined ones, and larger at the deepest layers again. And because we are not only interested in topography, but also in function, we also wanted to measure the spatial frequency tuning curves non-invasively using fMRI. So basically, we measure the maximum bold response to each one of these spatial frequency gratings, and here we are plotting this bold response as function of spatial frequency. And then we can fit one-dimensional Gaussian in which the center corresponds to the optimal spatial frequency and the width to the broadness of the tuning curve. And what we saw is that the optimal spatial frequency that we derived using fMRI is actually very well aligned with what we can measure using electrophysiology and calcium. So I hope so far I have showed you that the measures that we are measuring using fMRI are actually very biologically meaningful and accurate. And once we have this in mind, we went ahead and we studied how function measured via bold was altered between the healthy controls and visually deprived animals. And the first thing that popped up is that it seems that activation is much more enhanced in visually deprived animals when they saw light for the first time compared to healthy controls. In particular, we have a 3 times larger bold response compared to healthy controls. Although it seems that also the dark-reared animals respond more vigorously in LGN and superior colliculus than healthy controls, this was not statistically significant. But the most important factor is that we saw that in just 1 week of light exposure was sufficient to actually make the values in the visual cortex to return to baseline levels. Then we went ahead and we studied how this visual deprivation actually shapes the topography of the visual pathway in rodents. And while we can see that in healthy controls we have this very orderly shaped retinotopic organization, we see that the dark-reared animals, they have a total absence of this retinotopic structure. And one way that we had to actually quantify this is if we take that the definition of retinotopy is that neighboring neurons respond to nearby positions in the visual field. By computing the variation in receptive field sizes across cortical distance, we can see that the dark-reared really show that neighboring neurons respond to much sparse locations in the visual field. And then we looked how actually a prolonged visual stimulation shapes this reorganization. And the interesting point is that we saw that the retinotopic structure in dark-reared animals is progressively becoming more retinotopic organized. And at 27 days of prolonged light exposure are sufficient to result in very similar retinotopic maps between the dark-reared animals and the healthy controls. Then we wanted to look what could be the underlying mechanism of this specialization, and we looked at the receptive field sizes. And what we see is that in the visual cortex and superior colliculus, we see that at time point 0, the dark-reared animals really showed to have a much larger receptive fields than healthy controls. And the most important characteristic that is present throughout the entire visual pathway is that from time point 0 to time point 7 days, we actually see this shrinkage in the receptive fields that is then sustained over time. So there is this specialization of the receptive fields that is triggered by visual exposure, and that we can see actually in this map of specific voxel being tracked across the 4 time points. Just to show you that these results are not just at the topographic level, but actually at the functional level, we saw how we analyzed how the spatial frequency tuning curves are actually shaped by visual experience. And what we saw is that while at the first time amount of visual exposure, the dark-reared animals really showed to have a very broad spatial frequency tuning curve, so they respond to a broad range of spatial frequencies. 1 week after light exposure, there is a sharpening of these spatial frequency tuning curves, which is very well identified by this reduction of the width of the tuning curve. So to sum up, I hope I showed you that the adult rat visual pathway is actually very plastic, both in terms of visual deprivation that can delay the maturation of the visual system, and most importantly, that visual experience post-critical period can actually promote this refinement of the receptive fields and large-scale topography of the visual system. And if you would like to know more about visual plasticity in both rodents and humans, please check out my posters. Thanks a lot.
Thanks so much. We have time for some questions. Are there any questions from the audience? Yeah.
Thank you for the great.
Yeah.
Thank you for the great talk. Sorry, I can't use my app. It doesn't work well. But I was wondering, as someone who does not do that much research in this field about visual cortex and everything, but I heard that in blind people, usually, there is some plasticity that makes like the use of hearing, I think, of the idea start using their visual cortex to do some hearing processing or special processing linked to the hearing. Maybe I'm not exactly correct, but I wanted to know if you think you could test if this kind of thing happened in the rats or the mouse that are not being exposed to light at all, and then actually you could see a decrease in this kind of thing when you start to show them light and to actually stop the
To see if I understood correctly, your question is whether we see if the the auditory system has underperformance once the animals start to see again, or is it not that?
Somewhat. It's more. Well, maybe I'm completely wrong about my assumptions.
So we looked a lot at this multisensory integration. So we could also visually deprive the animals, and we could also place them in the space without sound, and let them grow without as close as possible without sound, which is much more trickier than without vision, because it's harder to cancel the sound, and the fact that also scanning makes a lot of noise. It's also very hard to scan it to show visual stimuli without sound. But we didn't see that so much of this cross-modal plasticity. So we do see changes in the amplitude of the bold responses, and that, for example, visually deprived animals have enhanced bold responses to auditory stimuli. But we didn't, in particular in the on this region of inferior colliculus of the rats, but we didn't see, this large scale. We didn't see, for example, that presenting stimuli would lead to in the terms of tonotopy to activity there, So. Yeah, so we were actually a bit disappointed. So we don't see so much this cross-model plasticity, at least in the models that we are testing. But might be because it's very hard to control actually the auditory stimulation throughout development.
Okay, thank you very much.
Okay, thank you. So in the interest of time, we'll move to the next speaker.
Okay, our next speaker is actually a recording. So Xiaojing Hao from the Department of Curriculum and Learning Sciences in Zhejiang Province, China. And their title is The Neurocognitive Mechanism of Processing Embedded Structure Within Block-Based Codes for Novices.
Hello everyone, this is Xiaojing Hao from Zhejiang University. The topic of my presentation is the neurocognitive mechanism of processing embedded structure within block-based codes for novices. In the 21st century, coding ability has become an important technological skill. However, novice learners always face many challenges to learn coding, one of which is the difficulty in processing embedded structure, which is defined as placing one control structure inside another. Previous studies have consistently found that novice learners prefer to use sequential structure that organize the two or more control structures sequentially to solve problems. Although such structure is more longer and resident. Such findings indicated that novice learners have difficulty in processing embedded structure. However, so far, the cognitive neural mechanism underlying embedded structure processing still remains unclear. To provide new insight into this question, we draw on the hierarchical complexity theory. According to this theory, embedded structure and sequential structure differ in hierarchical complexity. Specifically, as shown in the figure, embedded structure organizes the commands across more hierarchical levels than sequential structure. And the execution of code commands involved in the embedded structure requires much more iterative switches between the repeat control structures that are organized at different hierarchical levels. A lot of previous studies have provided neural and behavioral evidence for the involvement of cognitive control in hierarchical task processing. For example, as the neural basis of cognitive control, the frontal parental regions have been consistently found to be involved in hierarchical task processing. In addition, hierarchical processing has also been found to be supported by the functional connectivity between the basal ganglia and the frontal regions. Therefore, in this study, we assumed that the greater hierarchical complexity involved in the embedded than sequential structure would lead to greater demands on cognitive control to maintain and update information. Neurologically, processing embedded structure would lead to greater brain activity at the frontal parietal regions to maintain information, as well as greater brain activity at the basal ganglia frontal regions to update information. To test this assumption, we designed a code comprehension task that included the embedded and sequential conditions, with 2 or 4 commands inserted into each condition. Then we conducted block analysis to examine the difference in reactivity between embedded and sequential conditions. In addition, we further distinguished the commands involved in the embedded and sequential structures as transient and stay commands. Specifically, transient commands refers to commands that are placed at the boundary of the repeat control structure. After executing transient commands, individuals need to update more contextual information, such as how many times the control structure has been repeated. Then we conducted event-related analysis to examine the brain activity elicited by the transition and the state commands under embedded with sequential structure. As for the block-based code comprehension task, the code commands were designed to instruct the transformation in stimulus in shape, weight, or letter dimension. Corresponding to each code command, a pair of essential target stimuli only differed in one dimension was successfully displayed. During MI scanning, participants were required to follow the execution rules of embedded and sequential structures and judge whether the transformation from the essential to the target stimulus was consistent with the code commands. Block analysis results indicated that processing embedded with the sequential structure elicited greater activation in frontal parietal regions, such as the left superior and middle frontal gyrus, as well as the right superior and inferior parietal lobes. These results indicated that greater proactive control was involved to maintain information in embedded structure. Event-related analysis results indicated that, compared with sequential structure, the processing of transition commands within the embedded structure elicited greater activation at the basal ganglia frontal gyrus, such as the bilateral superior middle frontal gyrus, right inferior frontal gyrus, right pituitary, and left thalamus. Such results indicated that processing embedded structure requires more reactive control to update information at the boundary of embedded control structure. In conclusion, results from this study indicated that the processing of embedded with sequential structures recruited greater cognitive control, which resulted in greater activity in frontal parietal regions and basal ganglia frontal regions. As a result, the difficulty in processing embedded structure may stem from the greater demands imposed on cognitive control to maintain information proactively and update information reactively. That's all. Thank you.
Unfortunately, we can't ask questions of her, which is too bad because that was interesting. Do you want to introduce the next speaker?
Okay, so our next speaker is also unfortunately not here, so we're going to play their recording. So the next speaker is Shaheen Tavakol from McGill University. The title of Shaheen's talk is An Integrated Approach for Assessing Relational Memory in Healthy and Epileptic Individuals.
Hello and welcome to my presentation. My name is Shaheen. I'm a last year PhD student working at the Multimodal Imaging and Connectome Analysis Lab at the Montreal Neurological Institute. My work focuses on relational memory in humans, both in health and in disease. Relational memory consists of cognitive processes that bind the various elements of subjective experience. These include episodic memory, in which autobiographical events can coalesce into distinct episodes, semantic memory, where concepts can merge into general categories of meaning, and spatial memory, where the location of objects in space in relation to other objects can converge to form what is typically known as a cognitive map of the environment. Modern neuroscience lacks a standardized and holistic framework for the multidimensional assessment of these different types of relational memory. And in the age of big data and multimodal imaging and multivariate statistical analyses, I just had to do something about that. And so was born the Integrated Relational Evaluation Paradigm, or simply IREP. In this presentation, I will discuss how we leverage the IREP in conjunction with complementary multivariate statistical approaches to examine the behavioral difference in relational memory between healthy individuals and patients diagnosed with temporal lobe epilepsy. We examined 20 TLE patients and 53 age and sex-matched controls. All participants underwent multimodal MRI neuroimaging, including structural sequences, in addition to task-based acquisitions during which the IREP was administered. The IREP is a user-friendly Python-based program composed of 3 complementary modules: the episodic task, the semantic task, and the spatial task. The episodic task is administered in 2 separate runs, one encoding and one retrieval. During the encoding phase, pairs of images are shown at each trial. Half are displayed twice and the other half only once, as you can see. Thus, duplicate trials account for the easy condition, since the second exposure facilitates memory encoding, and single trials account for the difficult condition. During the retrieval phase of the episodic task, an object will appear at the top of the screen and 3 objects will be displayed at the bottom. It is the job of the participant to select the item that was originally paired with the object at the top. Unlike the episodic task, the semantic module is administered in a single run and does not have an encoding phase. Once again, an object will appear at the top of the screen with 3 options below. The participant must now choose which option is conceptually most alike to the top item. Here too we have an easy condition in which the correct response is fairly obvious, and a difficult condition in which it is a little less so. The spatial module also comprises distinct encoding and retrieval phases. But unlike the episodic, the spatial task is administered in a single run. At each trial, the participant must first encode the spatial arrangement of 3 objects. Within the same trial, the participant will have to select the same encoded configuration among 3 options. In the easy trial, the geometry of the correct spatial arrangement differs substantially from the other options, whereas this difference is minimal in difficult trials. We calculated percent accuracy scores for each condition in each task. Here the results are displayed for both cohorts, healthy controls on the left, TLE patients on the right. Red box plots correspond to the difficult condition in each task, and yellow box plots correspond to easy. We used linear mixed effects models as a starting point with task accuracy as the outcome measure. Our model consisted of a group factor with 2 levels, healthy control and TLE, a task factor with 3 levels, semantic, episodic, and spatial, a condition factor with 2 levels, difficult and easy, interaction terms for task and condition, and also group and task, and a single random subject term. We decomposed significant interactions and, where appropriate, ran post-hoc pairwise comparison tests while controlling for the false discovery rate. To provide additional context for the behavioral differences across groups, we implemented partial least squares correlation to determine if performance on the IREP was associated with specific demographic and clinical phenotypes. These features were age, sex, presence of TLE diagnosis, as well as left and right hippocampal volumes. Following linear mixed effects analyses, we were able to ascertain systematic differences in how participants performed as a function of task condition. As you can appreciate, accuracies were significantly higher on the easy condition relative to difficult, regardless of the task. Furthermore, semantic scores were much higher than either spatial or episodic, and this was true for both conditions. On the easy condition only, overall performance was better on the spatial task relative to episodic. Healthy controls outperformed TLE patients only on the episodic module, and while both groups scored highest on the semantic task, whereas healthy controls performed poorest on the spatial task, TLE patients found episodic to be most challenging. We observed that the first latent variable obtained via the decomposition of the cross-correlation matrix between clinical phenotypes and IREP accuracies accounted for a whopping 85% of the total covariance, and the correlation between latent scores along this first component was also evidently quite notable. To test for the significance of the first eigenvalue, we ran 5,000 permutations resulting in the present null distribution, against which the original singular value, which is 0.82, was compared. As you can see, the result was highly significant. We also applied a bootstrapping procedure with 5,000 iterations to assess the reliability of clinical and IREP loadings on the first component. This slide is very telling in that it shows that younger age, the absence of TLE diagnosis, and larger left and right hippocampal volumes collectively make up a phenotypic profile that, irrespective of sex, is associated with better multidimensional relational memory performance. In this work, we wanted to show that it is possible to examine the different dimensions of relational memory all at once and across different population cohorts. And we did this by leveraging a novel and holistic framework known as the Integrated Relational Evaluation Paradigm, or simply IREP. We use the IREP in conjunction with multivariate and data-driven statistical approaches to evaluate the differences in behavioral performance across healthy controls and patients diagnosed with temporal lobe epilepsy, or TLE. We found that performances were highest on the semantic module relative to the other two tasks. The main difference between groups was seen only in the episodic task, and the healthy controls clearly outperformed TLE patients in that module, we were also expecting to see similar findings in the spatial task, but given the relatively small sample size of the TLE cohort, examining larger groups may yet substantiate this expectation. Moreover, we also found that being relatively young and healthy and having relatively large bilateral hippocampi were also indicative of better behavioral scores across all the IREP outcome measures. I hope you enjoyed this presentation and ask me anything.
Okay, great. So maybe people can send him an email and compliment his mustache and also ask him some questions. Okay, so I'd like to invite the next speaker to the stage, Yoram. So Yoram Soch is from the Bernstein Center for Computational Neuroscience in Berlin, Germany. The title of his talk is Structural and Functional MRI Data Differentially Predict Chronological Age and Memory Performance. Let's welcome Yoram.
Yeah, thank you very much for the introduction. So yes, we're having a look in this talk, we're having a look at structural and functional MRI and their predictive value for chronological age and memory performance. So our research starts from the observation that although cognitive performance generally declines with increasing age, as is here shown for memory performance of older compared to younger subjects, some older adults typically show memory performance comparable to that of young subjects or even above the average of young subjects. And these subjects are commonly called successful ages. Now, it is still to be revealed what makes successful ages so successful. So one possible explanation is that these subjects simply have higher brain structural integrity as compared to other subjects who are already more strongly suffering from age-related brain atrophy. This could be called brain maintenance. Alternatively, these subjects might simply recruit the existing resources more efficiently or employ compensatory cognitive strategies. And this could be called cognitive reserve. Now, although we will not strictly rule out one of the two theories today, we can at least provide some evidence for them by testing the predictions that these theories make. Because according to the brain maintenance view, differences in memory performance within the older subjects should concur with differences in structure MRI patterns. Whereas according to the cognitive reserve theory, differences in memory performance should correlate with differences in functional MRI responses. So we measured functional MRI responses for a very large cohort of young and older subjects who were presented with novel images which were new to them at the time of presentation and pre-familiarized images which they had seen before, here called the master images. In a subsequent memory test, the subjects were then presented with all the old images from the encoding session, as well as a couple of new images, and were asked to rate these images on a 5-point Likert scale, ranging from 1 for this item is definitely new to me, up to 5 for I've definitely seen this item in the encoding session. Now, this allows to categorize encoding events based on the retrieval responses and allows us to infer on fMRI activity differences related to novelty processing by contrasting novel with master images. This is here shown on the left, and subsequent memory effects on the fMRI activity, which is here shown on the right, by contrasting remembered with forgotten items. So these two memory-related processes have rather similar neural correlates, And in previous work, we have shown how from these fMRI contrasts, one can calculate summary statistics or single value scores, which for given older adults indicate how strong it is deviating or how similar this subject is with the prototypical activation seen in young subjects. So one of these scores, the so-called fate score, the average activity within a predefined brain mask of memory-related brain regions is subtracted from the average activity outside this mask. We have developed an improved version of the score called the same score, in which we first are directly operating on the activation patterns of the young and older subjects. Second, also incorporate deactivations on the respective contrast. And third, also take into account the variability of the young subjects used as a reference sample when calculating how similar a given older subject is with the fMRI-related memory activation in younger subjects. So, we have fMRI contrasts indicative of novelty processing and subsequent memory. From these fMRI contrasts, we can calculate single value scores. We are also analyzing the behavioral data from all subjects, that is, how often they responded 1 to 5 for the old versus new items. And additionally, from each subject, we acquired a structural MR scan from which we extracted voxelwise gray matter volume. And we acquired a resting state fMRI session from which we extracted voxelwise mean percent of amplitude fluctuation, or M/RF in short. So this is a general measure of the amplitude of the signal under the resting state. We then used these feature sets to predict a number of target variables, namely age group, that is, whether a subject was young or old, chronological age in years, as well as memory performance measured as area under the curve in the fMRI subsequent memory test. We used support vector classification and support vector regression with tenfold cross-validation to predict discrete and continuous variables of interest. Okay, so first we assured that age group can be classified based on all these variables. And here we are already seeing some kind of hierarchy from behavioral over functional up to structural information, which most strongly allows to make this classification. If we're then looking at the continuous variables and focus on the older subjects, we see that chronological age within the older subjects is best predicted from structure MRI and resting state fMRI, here shown in green and cyan. However, these two have absolutely no predictive power for memory performance within the older subjects, which instead is best predicted from the fMRI memory contrast and from summary statistics calculated from the fMRI contrasts, here shown in blue and magenta. Okay, so we wanted to follow up on this finding and therefore partitioned the older subjects relative to chronological age and memory performance, so giving us old and young older subjects and older subjects with lower versus higher memory performance. And from each of these partitions, we then took the uppermost quartile and the lowermost quartile to perform a two-group comparison within the older subjects. So when doing this, we found a double dissociation between memory performance versus chronological age on the one hand, and structural versus functional MRI on the other hand. Meaning that when partitioning older subjects by age, there are no significant effects with respect to functional MRI, but strong differences in gray matter volume. Whereas if we are partitioning older subjects by memory performance, there are no significant effects in structural MRI, but robust differences in memory-related fMRI activity. So, to sum up, we found that chronological age is best predicted from structural MRI, but functional MRI is the best predictor for memory performance. Second, using just single value scores calculated from fMRI contrast is just as good or even better in predicting memory performance than taking the whole brain fMRI contrast map. And third, so taken together, our results suggest that superior memory performance in healthy older adults is better explained by more efficient recruitment of the memory networks rather than preserved brain structure, which would be indicated in structural MRI. So with that, I want to say thank you. And if you want to know more, please come to my poster. It's MT738, still being presented today. And if you want to know even more, please also see these other posters from my group members or from peoples from the lab, which I also belong to, where we're taking a closer look on A, the voxel-wise relationships between structure and functional MRI, and B, the neuropsychological correlates of the single value fMRI scores. So thank you very much.
Thanks for that nice talk. Do we have any questions from the audience?
Do you think that, you saw that the gray matter volume, for example, is not predictive of the memory performance? Sorry. You said that, we saw that the gray matter volume is not predictive of the memory performance, but I was wondering if you think that if you looked into another structural information, like, for example, myelin content, cortical myelin content that can be, predictive of the efficiency of the information integration, you could have seen and you could have, come up with some predictive power of that structural measure for the memory performance in older age.
Yeah. So we don't have data to check that because we didn't measure this, but one could speculate that although whole brain structure MRI map does not allow to predict memory performance, maybe due to issues of overfitting, that the volumes of individual regions may allow to do that. We have not found correlations with hippocampal volume, for example, in the sample of healthy older adults, I should say. So when we are correlating the hippocampal volume, left or right hemisphere, with the single value fMRI scores, which are themselves predictive for memory performance, we found no correlation in any of the pairwise comparisons.
Any more questions from the audience before we go to the app? Go ahead.
I have a question. So I guess the brain maintenance idea, you could think of it as structural maintenance, but it could also be functional maintenance, right?
Yep.
That you would show a brain activity pattern that's more similar to young people in high-performing older adults. And I might have missed this. Was that in there in the data that it was if you were more similar functionally to young people that you had higher memory performance in the older adults when you were calculating your difference score?
Yeah. I was here contrasting cognitive reserve and brain maintenance rather strongly in order to have a nicer introduction to the talk. But you're right that brain maintenance can also refer to functional maintenance or a maintenance of functional processing of information. And I would say that our results indicate that those subjects which score higher on memory performance are not characterized by fewer structural differences from the young subjects, like preserved hippocampal structure and so on, but rather by higher functional similarities with the young subjects. And that's what's indicated by also these single value scores.
One more quick question from the app. Yep. So do you think that the association between fMRI and memory performance could still be mediated by structural differences?
Could be. Question is, how can we test that? The idea would be that local gray matter volume is a mediating factor on the correlation between, for example, fMRI summary statistics and memory performance. Yeah, we could run a mediation analysis to check this. Thank you for the suggestion.
Okay, so I think we'll move on to the next talk. Thank you so much.
Thank you again.
Okay, our last speaker is Chiara Rossi from the University of Brussels. Title of her talk is A Unique Temporal, Spatial, and Spectral Description of the Working Memory Oscillatory Dynamics.
Thank you. Hi, everybody. My name is Chiara Rossi, and I'm a PhD student working at Ames at the Vrije Universiteit Brussels. And today I will present the first work of my PhD, actually, which consists in a comprehensive network analysis of the working memory brain dynamics. Okay, so working memory, as we know, is that cognitive function that allows us to remember an item and then recall it just after a few seconds. And the question that we investigate is, how does the brain perform this cognitive function? How does the brain dynamics underlying working memory look like? So what we know from the literature is that the brain dynamics, while performing brain dynamics, the brain recruits large-scale brain networks. These brain networks have long-distance brain regions that synchronize to communicate using different brain rhythms, such as theta, alpha, and so on. Also, what we know is that these networks wax and wane over milliseconds. Here I present the work of a former colleague of mine, Lars Kosters. He has conducted one of the most thorough and detailed investigations of the working memory brain activity. And he analyzed magnetoencephalographic data acquired during an NBAC task. But he applied methodologies that are quite traditional. So he performed an event-related field analysis. Can you use this? An event-related field analysis, which we know has not yet been proven to be very successful in revealing the brainwaves in MEG data. And then he also performed a time frequency analysis. Now, thanks to the MEG data, he managed to unravel the milliseconds temporal evolution of the brain activity, more specifically the theta, alpha, and beta power dynamics in source space reconstructed data, because MEG, as we know, it's quite easy to source space reconstruct. Okay, but there is a lot of information in this study. It's sometimes difficult to interpret and integrate, and additionally, the network information is completely neglected. So we decided to move forward, and we decided to conduct a dynamic network analysis on the same data set. And to do so, we have chosen to apply the time delay embedded hidden Markov model, which is an unsupervised learning model that is developed in the Bayesian statistical framework. And it has already been applied to analyze MEG data both in resting state and in task data. So what the model does is actually infer a predefined number of brain states that can describe the transient burst of activity throughout, in our case, an MEG recording. And every state is characterized by a spatial map, which is the power spectral density distribution over the brain, and then a connectivity map, which describes how the brain regions are synchronizing. Now, both these two maps are actually strictly related to a frequency range, as you can see here. Now, every frequency range then will yield to the specific function that we will associate to every state. Additionally, for every state, we have a time course, and the time course is actually the probability of activation of the state throughout time, over time. And what we can do in task data is to epoch the time course of every state with respect to the task information. And this will then give us to the pattern of activation of every state throughout time. Now, this is very nice, so we decided to perform this exploratory analysis where we conduct a network analysis by applying the HMM model to analyze MEG data. So the goal is to decode the time, space, and frequency dimensionality of the brain networks underpinning working memory. So, as I mentioned, we use magnetoencephalographic data. We use a cohort of 38 healthy subjects, and the data were acquired during an NBAC task. This is one of the most widely used NBAC working memory paradigm, and it consists in showing a sequence of letters. And the subject has to respond to a letter, a target letter. And the target depends on the paradigm condition. So it's the letter X in the 0 back, in the 1 back, the target is every letter that is identical to the one just shown before. And in the chew bag, the target is every letter that is identical to 2 letters shown before. So the MEG data was pre-processed using the OSL library. And it's worth mentioning that we source reconstructed the data using a beamformer, and we used a 42 regions parcellation. Then we applied the time delay embedded hidden Markov model to infer 6 brain states. Now, when we do model inference, we concatenate all the subjects and all the data, so we do not give the information about the task to the model. So the model is completely blind to the task information. Then, of course, we infer the brain states, and we extract the space map by mean power spectral density distribution of the brain, the connectivity map, as I mentioned earlier, and the time domain, the temporal course over the epoch. To do so, we epoch, as I said, the time courses of the states with respect to the task. We perform an ERF analysis by applying the GLM model. And here I propose the constant regressor of the GLM model. Now, the constant regressor is the mean activity common to all the paradigm condition. And as we see, there are 4 states that are significantly activated or deactivated throughout the epoch, which means that they specifically characterize the brain dynamics underlying working memory. And for the sake of this presentation, I will only mention 3 of them. So the first state that we observe is actually a state that peaks around 200 milliseconds. It's a very early state that activates significantly regardless of the paradigm condition. And this state is characterized by the power is focused in the prefrontal regions, as well as some, I don't know if you see, but some parietal regions, such as the posterior cingulate cortex, as you can see here. So both the connectivity map and the spatial map, they are referred to this frequency range, which is characterized by low frequencies, and we often associate this frequency range to theta activity. So because of the excitatory theta activity and the prefrontal regions involved, we likely think that this state is associated with executive control of stimulus encoding. And we were expecting to find this state because it's a signature of working memory brain dynamics. The second state we observe is actually a deactivated state, a state that significantly deactivates between 250 and 500 milliseconds. And this state is characterized by alpha activity in the occipital regions. As we can also see in the connectivity map, these regions are synchronized in the alpha power. But the deactivation of this network implies that these regions are actually desynchronized in the alpha range. So most likely, the deactivation of this network implies that these regions are locally involved in, for example, letter processing and memory storage and update. The last state that I propose, it's a quite special state. If you look at the pattern, the activation pattern looks like an M300, and it's almost the first time that we can see an M300 wave in MEG data. This state significantly activates between 250 and 500 milliseconds as well, even later, but its activation is significantly higher and longer in target trials rather than the structure, so when the subject has to respond. This state is characterized by broad frontal regions, as well as some supramarginal and sensory motor cortex regions, and the activity is associated with beta frequency band. As you can see, the connectivity map is quite broad, so to interpret this state and localize it in the working memory literature, it is quite difficult, but because of the beta activity and the involvement of supramarginal sensory motor cortex, we associate this state to stimulus manipulation and response selection. So to recapitulate, we try to decode the brain activity underlying working memory, and we try to do so with a very, very fine temporal resolution. We, in fact, managed to decode the network dynamics in an epoch that is shorter than 1 second. So after the stimulus presentation, we observe the arousal of a prefrontal state characterized by theta activity, which is associated with executive control of stimulus encoding. Then there is inhibition of the alpha conductivity in the occipital regions, which likely reflects local processing. And then eventually we have these M300 states that arise, which we associate to response selection and manipulation. But if anybody else has suggestions on the interpretation of this state, is more than welcome. The idea of performing this data-driven network analysis is also to then observe how these networks, so the time, space, and frequency dimensionality of these networks is then disrupted in subjects that are that have cognitive impairments, such as in patients with multiple sclerosis. Okay, so I thank you very much for the attention. This is my group. I want to thank them all, and yeah, that's it.
Okay, well, we've got a question that a couple of people are asking. So how did you determine the number of states?
Yeah, so it's a little bit of a few things that come along to define the number of states. So first, we look at the literature, and in HMM, especially in task data HMM inferences, the number of states is usually around 6, 8, but not larger. Also, then what we saw is we investigated the free energy. So we ran the inference, we ran the model a few times with a different number of states, and then we saw how the free energy behaves. And we saw that, of course, by increasing the number of states, we increase the free energy decreases, but then the complexity of the model increases exponentially, so it's very difficult to interpret the states. Also, what happens is that the higher the number of states, then the higher is the information that some states share, and so there is some kind of redundancy within states. So basically, eventually, we saw that 6 is quite a good number of states to explain the brain dynamics in such a short epoch. I also have the results of 8 states, but it doesn't say anything more. It doesn't say anything interesting anymore. So yeah, that's a little bit the chart. More, a little bit methodological, a little bit of reasoning. So yeah.
Maybe related to that. So how do you get the frequency from each state?
Yeah, so, well, okay. So we calculate every state has a power spectral density distribution, so that's it. And then from that, we ran non-negative matrix factorization. So from that model is actually extracting the frequency content in every state. And then you give in to the model also the number of modes, frequency modes. I can go back. So in our model, we infer. I'm sorry, it's a lot. So here, for example, this is the paper by Quinn. I report the figure, but so we have 4 modes that we want to extract, so 4 frequency ranges that we would like to extract. And the non-negative matter factorization algorithm, what it does is to extract basically the frequency content from the power spectral density distribution of the states. Yeah.
Okay. Anyone want to ask a question, like in person, talking? No?
There's a hand over there.
Oh, there's a hand. Hey, there's some hands. Thank you.
Hello. So you said that the beta state might be related to motor activity or something like this, right? Engaging in a response.
Well, I don't classify that state as a motor state.
Okay.
Because it doesn't involve also sensory motor regions, but it involves also a broad frontal.
Sure.
So I don't classify it as a motor state.
Okay, but I wonder, so everything is time locked to the stimulus onset?
Yes.
I wonder if you time locked it to the response onset and looked backwards from there to see if this beta thing is happening there.
That's actually a great suggestion. Thank you very much. It could be definitely a good suggestion. And that's also one of the great advantages of having such a nice temporary solution, because then we could definitely run this analysis, because we see the mean reaction time is really within the window of observation. So thank you very much for the suggestion.
No problem.
Okay, one final question that's been upvoted. So did you find any relationship between the states and behavioral performance on the task?
So it's something we're looking into. Actually, tomorrow we'll present another poster on where I applied this model to see how the states change in patients with multiple sclerosis. And there we try to relate the peak of activation of every state with the cognitive performances of the patients. We haven't seen interesting correlations, but what we have seen is that the peaks of activation, especially for the prefrontal state, does significantly vary between patients with multiple sclerosis and healthy controls. So we do see that in patients with multiple sclerosis, the peak is smaller, and also this peak is delayed. So we do see interesting results, and we do see potential for this methodology to explain us a little bit better how cognitive impairment, especially in working memory, work. Yeah.
Okay. All right. Thanks very much. Thank you to all our speakers, and thank you for coming. We survived. Yeah. Thank you for writing all your. Yeah, nice talk.
Yeah.
Oh, no, yeah. No. We were just like, is he here?
Yeah,
and we
sorry,
don't know he's here.
We didn't know how long the videos were. If we had known that they were going to be short, we would have given you another question. But because the videos were a little short, so.
Thank you. Good job.
Yeah.